So, good morning. We will be spending today's activity extracting DNA from fruit. So, first of all, be careful, use caution in this activity. Do not misuse, digest, or play with anything used in the activity unless instructed that it's okay. The banana DNA and the mixture surrounding it, or fruit DNA, is not safe to consume. Do not try eating it after you've worked with it with chemicals. So identify the materials needed for this experiment. I'll tell you what you need for the experiment, but most of the supplies you have already in your science box. Some of them, like a jar or things like that to mix things in, those things you'll need to find at your house. I've extended the due date on this assignment to next week just because one of the things you have to do first is find in your science box the alcohol eyedropper, the isopropyl alcohol. It's rubbing alcohol. It's the bottle in your box that's marked with marker, with black marker. And you need to take that and put it in your freezer. The experiment works best when that is as cold to as close to freezing as possible. So while you're getting your other materials done, put it in the freezer if you'd like. But essentially, you're making a science journal prompt. You can do this as a science journal prompt. Oh, excuse me. A science journal prompt through the digital copy, or you can do a worksheet, which is attached to this document as well. So the science journal prompt is this. What does the fruit DNA look like after you've worked on the procedure? And if you had difficulty with the process or something didn't work fully, explain the circumstances here. There's no failed experiments, just opportunities to be learning from this. What are some practical applications to being able to extract the DNA from something like what purpose would you have possibly? What do you think we can use DNA for? Like what kind of applications? Name 5 things that don't have DNA and then add a diagram, model, photo, or video of the process. So you have to document how you what you did in this experiment. Again, there's no failed experiments. This is just an opportunity to try and see how it goes. So if you click on this image below, you'll get a digital, you should get a digital journal entry. I'll switch these words around, but this one right here, this will take you to the procedure and materials list. So your materials that you actually need for this are, you need very cold rubbing alcohol, that's your eyedropper. You need a coffee filter, like a paper filter, or alternatively, a paper towel, a narrow glass to mix things around in, a wooden stir. The stick in your box is given to you for that. You will need a quarter of a peeled ripe banana, or, excuse me, or that much amount of strawberries or another fruit. It doesn't really matter what fruit you use, but bananas are easier to find in most people's houses, and it's kind of easy to work with because it's a squishy fruit. You need a quarter cup of hot water, half a teaspoon of salt, and a quarter teaspoon of liquid dishwashing soap, like Dawn dish soap or something like that, and a Ziploc bag, which is in your box as well. So after you've worked on the procedure, here's step 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's only 10 steps. And there is an explanation of kind of what happens here in extracting the DNA and a worksheet. These are the same prompts as in the description above. So you can either print or do this on the worksheet, or you can make your own science journal prompt as well. So clicking this image will give you just the journal prompts to type in your answers and stuff. Okay. So again, you can do this on paper, you can do it digitally, but I would like you to upload a photo or some sort of diagram or model or video of your process, because I'm interested to see what your results look like and kind of see your process along the way. Okay. If you need any help with this assignment, like how to do certain steps, please email me. I'll be more than happy to walk you through some of this. Good luck, and I will see you in Zoom.